Good morning, students. We have started the revision of fundamental unit of life, and already we have discussed so many things as part of revision. In the last class, we have discussed about nucleus. While discussing about nucleus, we have discussed about prokaryotes and eukaryotes. Prokaryotes, they contain nucleoid and they do not contain nucleus. Prokaryotes do not contain membrane-bound organelles also. Whereas at the same time, eukaryotes contain defined nucleus or membrane-bound nucleus and other membrane-bound organelles, cellular organelles or cytoplasmic organelles also. As prokaryotes do not contain membrane-bound cytoplasmic organelles, functions of such organelles are performed by poorly organized parts of the cytoplasm in prokaryotic cells. Hence, in the prokaryotic cells, there is no defined nucleus or membrane-bound nucleus, and there are no membrane-bound cytoplasmic organelles. Though the prokaryotes do not contain membrane-bound cytoplasmic organelles, functions of such organelles, they are performed by poorly organized parts. Poorly organized means no boundaries, but some specific regions are there, wherein whatever the functions that are carried out by cytoplasmic organelles of eukaryotic cells, same functions are carried out by these poorly organized parts of the cytoplasm in prokaryotic cells. For example, in the case of eukaryotic cells, there is a special organelle called plastid. Okay, if the organism is of autotrophic in nature, such organisms in eukaryotic cells, in eukaryotic organisms, if the organism is photosynthetic type, then in such organisms, special organelle called plastid is present. In photosynthetic prokaryotes, for all prokaryotes, example are bacteria. So in photosynthetic prokaryotes or in photosynthetic bacteria, the chlorophyll, it is present in membranous vesicles, whereas in eukaryotes this chlorophyll is present in plastids. Okay, in photosynthetic bacteria, chlorophyll is present in which part? In membranous vesicle. There is a lot of difference between membranous vesicle and a plastid. Plastid is a double-layered membrane-bound clear organelle. Vesicle means it is like a soup bubble, right? It is not having any tough membrane like that of a plastid. And chlorophyll as a pigment, it is simply present in that vesicle. Whereas in plastid, the chlorophyll is present in again special part of the plastid called grana. We will discuss about it in higher classes little in detail. Remember that one. In photosynthetic prokaryotes, that is in photosynthetic bacteria, chlorophyll is present in membranous vesicle, whereas in photosynthetic eukaryotic cell, chlorophyll is present in plastids. Now we are going to discuss about the fluid content of the eukaryotic cell, not of a prokaryotic cell, right? Before discussing this, let us discuss about complete differences between prokaryotes and eukaryotes. The differences between prokaryotic and eukaryotic cells. Prokaryotic and eukaryotic cells, they exhibit some differences. What are those differences? Prokaryotic cell, due to their poor structural organization, their size is very less or small. They are about 1 to 10 micrometer in size. Whereas eukaryotic cell, they are about 5 to 100 micrometers in size. The symbol is micrometer, right? And in prokaryotic cell, nucleus is absent and nucleoid is present. Whereas in eukaryotic cell, defined nucleus is present. In prokaryotic cells, only one chromosome, single chromosome is present. Whereas in eukaryotic cells, generally more than one chromosome are present. And in prokaryotic cells, membrane-bound cytoplasmic organelles are absent. Whereas in eukaryotic cells, membrane-bound cytoplasmic organelles are present. And since they are having poor cellular organization, prokaryotic cells are restricted to the level of unicellular organisms. You will not find a multicellular prokaryotic organism. All prokaryotic organisms are unicellular organisms only. Whereas in eukaryotic cells, in eukaryotes, there are both unicellular organisms and multicellular organisms. Because of the well-developed cellular organization, multicellular forms were developed from unicellular organisms. Because of the well-developed cellular organization, multicellular forms were developed from unicellular organisms in eukaryotic organisms. Now, let us discuss cytoplasm. The large region of cell that is enclosed by cell membrane and takes up very little stain during the staining process is referred as cytoplasm. It is in terms of biology laboratory. So when you are working in laboratory, someone is asking you what is cytoplasm. Then you can explain what is cytoplasm by showing this thing. What is that? The large region of the cell that is enclosed by cell membrane and takes up very little stain during staining process. You can call it as cytoplasm. But actually, theoretically, what is cytoplasm? The fluid content inside the plasma membrane is called cytoplasm. The fluid content inside the plasma membrane is called cytoplasm. And this cytoplasm, it takes up very little stain during the staining process. This cytoplasm takes up very little stain during the staining process in the biology laboratory. Cytoplasm, it contains the cellular organelles which perform specific functions for the cell. Cytoplasm, it is fluid, but inside this fluid, many membrane-bound cellular organelles or cytoplasmic organelles are present. And what is the role of these organelles? These organelles perform specific functions for the cell. And remember, organelles perform specific functions for the cell only in eukaryotic cells. Cellular organelles are enclosed by membranes. Many times we have discussed so far, cellular organelles are enclosed by double-layered membranes mostly. Having membranes enables the cells to show characteristics of life. By having plasma membrane, by having membranes surrounding the cellular organelles or cytoplasmic organelles, it enables the cells to show characteristics of life. It enables the cells to show characteristics of life. If you take some special creatures called viruses, they do not have membranes, means they lack membranes. So they do not show characteristics of life. So membranes containing creatures, whether they are prokaryotic cells or eukaryotic cells, they show the characteristics of life, true characteristics of life. Whereas the creatures or the organisms which do not have membranes or which lack membranes, they do not show the characteristics of life until they enter in a living cell or living body and use its cellular machinery to multiply. So viruses do not show any characteristics of life since they do not have any membranes. But they may show some characteristics of life like a multiplication. When when they enter in a living cell or living body, then only they show that multiplication property of living organisms. Otherwise, they do not show even that one also. If you take the large and complex cells, they need a lot of chemical activities to support their complicated structure. To support their complicated structure, large and complex cells, they need a lot of chemical activities. These chemical activities are of different types. These chemical activities are of different types. Okay. Cells use membrane-bound little structures or organelles to keep the different chemical activities separate from each other. We have discussed just now that these chemical activities are of different types, and these chemical activities have to be kept separate from each other in order to be performed efficiently. But how are they kept separately from each other? In order to keep the chemical activities separate from each other, cells use membrane-bound little structures called organelles. Okay. Some of the organelles can be seen with a normal microscope, and some organelles can be seen only through electron microscope. Examples of such organelles are Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. If these organelles are not there, then the needed chemical activities would not have been kept separate from each other, and the cellular organization would be in poor state, with which their large and complex cells they would not have sustained. Okay, so what are those organelles? How this Golgi apparatus, lysosomes, mitochondria, endoplasmic reticulum, vacuoles, all these, how are they going to function? All these things we will discuss in the next class as part of revision. Already we have discussed in the starting classes, but we will discuss once again the key points of such organelles. Okay, I forgot to write endoplasmic reticulum here. Let me write. So examples of some important cellular organelles are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. Okay, thank you.